Good morning, ladies and gentlemen. Congratulations on AGIL 30th anniversary. I wish you tremendous success in everything you do. Okay, today I'll talk about identification of gamma ray irradiated South Sea cultured pearl. The presentation outline is as follows. Pearl treatment, introduction, chemical composition of pearl, change of pearl by irradiation, detection of irradiation, irradiated South Sea cultured pearl, and conclusion. First, I'm going to brief you on the pearl treatment. Like many gem material, cultured pearl have a long history of accepted treatment. Japanese producers have developed a number of processes to improve the appearance of their pearls. The first step in traditional aqua processing, called maeshori, immersing the pearls in a solvent, usually methyl alcohol, for cleaning. It is now used to improve to luster. The next step in the traditional process is bleaching. Bleaching removes dark organic compound and creates pure white. After bleaching, some of pearls are treated with an additive to create the slightly pinkish overtone in many aqua pearls, seen in many aqua pearls. After bleaching, the final step is tumbling to improve luster. Sievage does not require that the steps in the traditional process be disclosed, but radiation and dyeing, bleach, heating, coating, and so on are requirements declared. They are widely used methods to change pearl color. These pearls were treated by irradiation, by bleaching, by dyeing. They belong here. It does necessitate the requirement declared about this treatment. Irradiation is widely used method to change pearl color. Until now, lower quality fresh water and salt water cultured pearls used exposed to gamma radiation to simulate black pearls or enhance orient. Most artificial dyed colors are easily recognized using gemmological testing, UV visible, and Raman spectroscopy. Also, most treated cultured pearls by high dose irradiation are easily recognized using gemmological testing. A few years ago, the irradiation process has been applied to not only aqua pearl and freshwater pearls, but also to South Sea cultured pearls with colors ranging from white to cream. Korean consumers prefer South Sea cultured pearl with a silver color created by organic material. We heard that some South Sea cultured pearls used to be treated with a low dose irradiation below 1 kilogram in these days. They were light gray or silver color. Unfortunately, it is very hard to identify treated pearl by low-dose irradiation. So, this study attempts to identify irradiated South Sea cultured pearls by low dose using ESR spectroscopy. Radiation can be simply defined as energy emitted in the form of particles or electromagnetic waves. There are three types of radiation: cobalt-60, electron, and neutron. Cobalt-60 is causative of gamma ray. Of these, penetrating power of gamma ray is very powerful. This gamma ray is being applied in various fields. For example, food, medical device, cosmetics, and so on. For this study, South Sea cultured pearls were exposed to radiation. The radiation was conducted in the cobalt-60 facility of the Korea Atomic Energy Research Institute in Korea. The experiments were carried out in twice. First, the observed dose was set at 0.5, 1, 5, 10, 30, and 100 kilograms. Second, the observed dose was set a low dose irradiation from 0.2 to 1 kilogram. Freshwater shell used in most cultured pearls today for nuclear bead. As you know, saltwater cultured pearls are composed of nacre and nucleus bead. After gamma ray radiation, both freshwater cultured pearl and nucleus bead in saltwater cultured pearl are darkening. It has been reported that the darkening of nucleus bead is due to trace MnCO3 oxidation into Mn2O3 or MnO2 by gamma ray radiation. This is the EDXRF spectra of freshwater and aquaculture pearl. The manganese element is found in freshwater cultured pearl. By the way, it is not found in saltwater cultured pearl. This table shows the chemical composition of South Sea cultured pearl using ICP AES. As shown on the bar graph, green bar means nacre and pink bar means nucleus. Nacre contains more natrium and magnesium and strontium, while the nucleus has higher manganese and phosphorus content. Pearl nacre contains aragonite plate. Various organic compounds occur between the aragonite, producing a range of pearl's color. Conchiolin exists between the aragonite plate. Conchiolin is organic protein. Protein is made from about 20 kinds of amino acid. This table shows amino acid composition of the pearl nacre before and after irradiation. The pearl nacre contains 18 kinds of amino acid. You can see the table. For a part of amino acid are destroyed after irradiation. glutamic acid destroyed from 3.5% to 3.1%. Alanine from 22.5 to 21.8%. And histidine from 1.6 to 0.9%. The destruction of protein by irradiation can bring about color change of South Sea cultured pearl. If the destruction of the human protein, what is happen? Probably like this. Also, such a thing in human being could be happen. We compare the change of nuclear speed and acquired pearls by irradiation dose. Before irradiation, the color of nuclei is white, and the color of acquired pearl is pinkish white. As the irradiation dose increases, acquired pearl becomes nuclear speed becomes darker, and acquired pearls becomes from pinkish white to bluish gray. Change the color of nuclear speed affect the appearance of the acquired pearl. This is the result of colorimeter. L is brightness, positive A is red, and negative A is green. Positive B is yellow, and negative B is blue. As you can see the graph on the left, brightness of nucleus has dropped sharply. You can see the similar result for the aqua cultured pearl. As the irradiation dose increase, the plotted point proceed toward the negative A and negative B. Also, we compared a change of nuclear speed and South cultured pearls by irradiation dose. Before irradiation, the color of nuclei and South cultured pearls are white. As the irradiation dose increase, nuclear speed becomes darker. But South cultured pearls are slightly changing. This might be due to the nacre thickness of the South Sea cultured pearl. If the pearl nacre is thin, the pearl color was changed remarkably to silver color. Identification of irradiated pearl by low dose irradiation. Why is it so difficult? There are two methods to identify irradiated saltwater cultured pearl with gemmological testing. One is observing the nuclear state through the drilled hole in the pearl. The figure on the left is aqua pearl and the right is South Sea pearl. After irradiation with 100 kilograms, the nucleus of aqua and South Sea were darkening. After irradiation with 1 kilogram, both of the nucleus change very slightly. It is very difficult to identify whether pearl has been irradiated or not. The other method is the light transmission. It's to check how much degree of transmitted light. The figure on the right, transmitted light is dark and shows orange color in high dose irradiated South Sea cultured pearls. In contrast, unirradiated or low-dose irradiated South Sea pearls are bright and look yellow-white. This is why detecting irradiation is difficult. Also, we checked UV fluorescence reaction on the long wavelength. The chalky bluish-white color was observed in the unirradiated South Sea pearl, but fluorescence was reduced as increasing dose. There are almost no difference below 1 kilogram. We cut the South Sea cultured pearl for observing of cross-sectional. The left photos are unirradiated pearl. The right photos are irradiated pearl by low dose 1 kilogram. There are almost no difference between them. It is not easy to distinguish which one is irradiated. So, how to identify? There are several physical detecting methods: PSR, TR, XPS, NMR, ESR, and so on. PSR and TR are generally used to detect irradiated food. In this study, it is suitable to identify irradiated South Sea pearl using ESR. ESR is a spectroscopic method for observing the resonance absorption of microwave power by unpaired electron spins in magnetic field. ESR can be used to detect paramagnetic defects or free radicals introduced by radiation. To identify if something is irradiated or not, it could be obtained through free radical of an radiation-related signal. To perform ESR, the nacre and nucleus bead were separated from South Sea pearl and then made them into powder. In our experiment, we used ESR attached manganese marker. Manganese marker is standard reference sample. It can be used to correct for any systematic errors and to verify the sensitivity of the system. The spectra show a South Sea pearls, nacre and nucleus bead before and after irradiation up to 1 kilogram. These spectra obtained a single pearl irradiated by different dose. Formerly, non-existent free radicals are found after low dose irradiation. G factor could be identified what kind of free radical. The G factor was 2.0015. So the free radical is produced by gamma ray radiation is CO2 minus. This Mn2 plus signal come from manganese marker attached to ESR. CO2 radical exists between the third and fourth Mn marker signal. The CO2 minus radicals appeared as irradiation dose increase both nacre and nucleus. Nucleus spectra show a large unassigned signal either before and after irradiation. So CO2 minus radical is easier to observe in nacre rather than in nucleus. The CO2 minus radical intensity of nacre and nucleus are normalized by unirradiated spectrum. The CO2 minus radical's intensity increase depending on irradiation dose. As you know from the graph, the CO2 minus radical is in nacre is more distinct than in nuclears. Mentioned before, the change of MnCO3 oxidation number affects irradiation-induced color change. Add to this, we found the color of nacre and nuclears had been bleached. This is irradiated nuclears by 30 kilogram irradiation dose, and is irradiated nacre by 10 kilogram, irradiated freshwater pearl by 10 kilogram, and irradiated aqua pearl by 30 kilogram. We tried to pay the test under incandescent light for 5 months. 5 months. The light temperature is around 50 degrees. After paid test, nucleus and nacre were bleached. Generally, the color change by radiation and light is related to the color center. Therefore, the color change of nacre and nucleus are related to color center. A sampling process for ESR. To set on the pearl jewelry, it must be drilled, make a hole. So we can get a powder just 10 milligram of nacre from the drilled hole for the sample. Because this method is not completely destructive, so we can tell this process causes non-destructive inspection. In conclusion, okay, that's the end of my talk. Thank you for your attention.
My question is, because we try to find an easy way to identify.
Yes, high dose radiation.
I mean, under the 200 magnification, did you see any difference on the surface before and after radiation?
Low radiation?
Yeah. That's my question.
It's no difference.
No difference.
Yeah.
The image.
Yes, only image, no difference. And we researched last year and early this year. There's so many irradiated South cultured pearl were trading in Korea pearl market, but now irradiated South pearl is decreasing. I think there's nothing wrong with treatment, but the seller must tell consumer about treatment about treatment at the point of sale. Disclosure is very important. Okay, thank you.